Anatomists, I wanted to go over what we'll be covering in the endocrine system lab. Now, this lab usually takes place in the first week of the semester, and you haven't had much information, if any, on the endocrine system. So I just wanted to provide a little bit of background. There's two main kinds of glands in the body. There's exocrine and endocrine glands. And the main difference between these two glands is where their secretions are sent. So exocrine and endocrine glands both produce things that are released. These may be hormones, they may be digestive juices, they can be all sorts of things. But the difference in how they're classified is where that substance goes after it's made. Exocrine glands will dump their products into ducts, and from there they can move on to body cavities or to the surface of the skin. So, for example, sweat comes from an exocrine gland. Digestive juices come from exocrine glands. Endocrine glands are different in that their hormones are secreted directly into the interstitial fluid that immediately surrounds the cell. So the cell makes this hormone, and then from the cell, it just goes right to the outside. So it doesn't move into a duct, although often hormones will ultimately move into the bloodstream. And so this is why when you're studying endocrine glands, especially if you're looking at the histology, very close up, highly magnified, you'll see a lot of blood vessels around. And this is because most hormones will ultimately move into the bloodstream. And organs don't have to be classified as only one or the other. The pancreas, for example, is both exocrine and endocrine in nature. The digestive juices that it makes move into ducts that make their way down to the small intestine, and it also produces insulin and glucagon, which are endocrine hormones, and those ultimately will find their way into the bloodstream. Endocrine glands will secrete their cells from epithelial cells, so glandular epithelium will be responsible for making these products. There's a plenty of connective tissue that is around for support. Remember, epithelium has no blood vessels, and frequently the hormones made by endocrine glands need to get to a blood vessel. And so you see a lot of connective tissue supporting that epithelium. And like I said before, endocrine glands frequently have a really great vasculature, extensive blood supply. Here is a quick overview of the different endocrine glands in the body. You can see there's some in the brain. Here's the pineal gland, responsible for producing melatonin. You have the hypothalamus and pituitary right here. The thyroid is found in the throat area right here. The parathyroid glands are very small glands located on the posterior or backside of the thyroid. The thymus is found just over the heart. You will not find a thymus on the adult models that we have in lab, and this is because the thymus atrophies a great deal after adolescence. So in children, you see a pretty large thymus, but in adults, it has atrophied a great deal and there's not much left. And so our adult models don't show very much of a thymus. They don't show any thymus. But when we look at the fetal pig, you'll see quite a large thymus sitting over the heart. The adrenal glands are these small glands that sit right on top of the kidneys. They're going to be retroperitoneal, so they sit behind the peritoneum. As you do your dissection, you'll need to move all of the organs out of the way that are sitting on top of these. The pancreas is nestled in between the stomach and the small intestine. And then the gonads, in addition to being reproductive organs, they are also classified as endocrine organs. This is a chart that appears in your lab manual. You need to know it. As we give you lab questions, one of the questions that we'll ask is to identify this endocrine gland or identify this particular cell on a microscope picture that we give you. And we may also ask you to identify a hormone or multiple hormones that are released from that particular cell or gland. So make sure that you know this chart really well. And we'll go over these hormones and their functions in lecture in addition to this. You also have a key to the models in your lab manual. Here are the list of the endocrine glands. Here is the number that is shown on the torso model. In this case, you see a blank box that's not grayed out. This means that you can see the hypothalamus in the torso model. It just does not have a number on it. You are still responsible for knowing this structure, even though it has no number. The torso models, as indicated by their name, only include the torso. They don't include the ovaries or the testes. You cannot see them at all. They are not present. And so that's why these boxes are grayed out. They are not present, and you are not responsible for knowing to identify the ovaries or testes on the torso model. We also have a full body muscle model. So you can identify the ovaries and testes on there, in addition to the rest of these glands, with the exception of the pineal gland. The pineal gland you cannot see on the muscle model, and you're not responsible for it. One of the main things that we're doing in this lab is beginning our dissection of the fetal pig. In your lab manual, you have a list of instructions, and your instructor will go over some of these with you. It's important that you collect all of the materials that you'll need. We'll have tools, gloves, dissection trays, and the pigs all available for you. Make sure that you come prepared to lab. So put away your backpacks. Make sure those are tucked under your tables. Make sure that if you know we're dissecting, bring hair ties, don't wear drooping clothing, all of that kind of thing. We also will provide bags for you to store your pigs in. At the end of each lab, please make sure that you do a good job cleaning up. Rinse and dry all of your dissection tools so they won't rust, and replace them back in their proper bins. Rinse off all of your dissection trays. Spray your pig with a little preservative and put it in your bag, and put your bag in the proper cabinet. Please make sure that you label your bag properly with all of your group members' names and the lab time that you are in. This will help us in case a pig gets misplaced. It just helps us to find it a little bit easier. If you have any questions about how to clean up your station, please feel free to ask a TA or your instructor whatever your question is. We're happy to help you. As you start dissecting for this lab, we use these pigs through the entire semester to look at all of the different organ systems. So it's important that you take care as you start cutting your pig. Make sure you do not cut any major blood vessels. Please take care not to puncture the heart or the lungs. We'll be looking at those later. Please be careful to not completely remove skin from the abdominal cavity. And if your pig is male, you can dissect out one of the two testes. Please also use care not to cut the umbilical cord off. We will look at the vessels in a later lab. Here is how your dissection will be a little bit different. The incisions you make will change depending on if you have a male or a female pig. The best way to determine if your pig is male or female is by the genital papilla. So over here, you can see there's a small triangular structure just under the tail. If you lift up the tail, that is the genital papilla. That indicates that your pig is female. Over here, you can see on the male pig, the genital papilla is absent. The presence of a scrotum or testes is generally not a reliable indicator of whether your pig is male or female. The genital papilla is the best way to tell. If you have a male, no genital papilla. This is a diagram of the incisions that you'll make. So you have one going all the way down the ventral or belly until you reach the umbilical cord. And as I said before, take care not to cut off the umbilical cord, but go around it instead. And then you have a few transverse cuts that you can make in order to open up the flaps of the cavity. If you have a female pig, it will be very similar, but instead of making two cuts to the side of the umbilical cord, You'll just make one incision that goes all the way around the umbilical cord, making sure that the umbilical cord remains attached. Here is a picture of what the incision will look like. So here's the liver. So the head of the pig is over here to the left, and we're zoomed in on the abdominal cavity. Here are the legs on this side and this side. Here is the umbilical cord still attached to this pig. You can see the urinary bladder is still present underneath. Make sure you don't cut through it. And this umbilical vein, it's a small vessel that runs from the umbilical cord up under the liver. Please take care not to cut this umbilical vein, since we will be studying it later. Here's another view of how those incisions are made. Here is the umbilical cord right here. Take care not to cut it off. And then everything in here, if you have a male pig, this all is going to remain intact. You can see these transverse incisions over here allow you to open up the flaps of the pig so that you can see all of the organs inside. Here's another image. You can move everything off to the side and still have good visibility while still leaving that umbilical vein intact, as well as your umbilical arteries and the urinary bladder right here. Here's the umbilical vein again and the umbilical cord. When you're looking for the pancreas in the pig, you'll need to lift up the stomach. So you can see the stomach right here is being lifted up. Here's part of the liver over here. This is the umbilical vein right here. These are small intestines. You can see the pancreas right here. It just looks like a chewed up piece of bubble gum to me. So the texture will help you identify this organ and separate it out from the small intestine, for example. So look for the chewed up bubble gum underneath the stomach when you're looking for the pancreas. The adrenal glands, as I said before, are retroperitoneal, and so they're going to sit on the back wall, the dorsal wall, behind all the other abdominal organs. So here's the small intestine here. You can see a little bit of the large intestine right here. Here's some of the liver, and you can see this peritoneum. So the parietal peritoneum is a membrane, and you're going to need to use a blunt probe to move through that in order to access the adrenal glands, which are quite small in the fetal pig. So this is your kidney right here. So you're going to be looking for something underneath this membrane right on top, right there. When you have that membrane removed, you can see this much better. So here's your kidney with the membrane removed, and right here, this little kind of grayish bean sprout looking thing right here, this is your adrenal gland. And then you can see the pancreas right next door. Up here is your liver, this is your spleen that's being picked up, and liver is off to the left. This one is your stomach right here. Down here, you can see the ovary. So this is obviously a female pig. Here's your large intestine, small intestine. Here's the kidney right here. This is your adrenal gland right here. You can see just a little bit of the pancreas right here. And ovary is going to be way down here. Ovary will be right next to a very convoluted tube, and that big convoluted tube is the uterus. And we'll go into some more of these structures when we look at the reproductive system. For now, you just need to identify the ovary. Looks like a small bean right next to convoluted tubes. As we zoom out a little bit, we look at the thoracic cavity. Now, as you are making the incision through the thoracic cavity, you're going to want to do it slightly off-center. So don't cut straight down the midline. You will need to cut through the ribs, and the cartilage that connects the ribs to the sternum is the easiest place to cut through. So if you cut just to the left or just to the right of the sternum, that will be the easiest way to cut through. And when you're using your scissors, not a scalpel, but when you're using your scissors, cut in an upward motion so that you take care not to accidentally pierce the heart. Once you've opened up that thoracic cavity, now you can see the heart and it's still covered in its pericardium, the sac that covers the heart. You can see the lungs off to the side. Here's your right lung and left lung. And then your diaphragm, that muscle involved in breathing right underneath. Here is the thymus, and you can see it's pretty large in the fetal pig. And that's this organ that sits right on top of the heart. You'll also find some thymic tissue in the neck of the fetal pig. And you can see that right here. So in addition to right over the heart, this area right here is also part of the thymus. Take care as you're doing these incisions and trying to isolate the thymus not to damage these large blood vessels, because we'll be studying those later. Here's another picture. As you move the thymus in the neck to the side a little bit, and you can just do that with a blunt probe. No need for a scalpel after you have the main body cavity open. The thyroid lies just underneath. Now up here is the thyroid cartilage, and it's kind of a good landmark for you. Thyroid cartilage is the enlarged cartilage that makes up the Adam's apple in men, and so it's a large distinguishing feature in the trachea, or just above the trachea. If you can feel that, your thyroid will be just caudal to that. So move on down, move the thymus out of the way, and this small, round, dark area, this is going to be your thyroid gland. But you will need to move the thymus to the side. If we look at the picture before, you can see the thyroid would be right around here, but it's hidden. So you have to move that to the side to expose the thyroid. And be careful on your quizzes and exams not to confuse thymus and thyroid. We're also going to be looking at some of these tissues under a microscope. The hypothalamus and the pituitary gland are often kind of lumped together. The pituitary's nickname is the master gland, but really it's the hypothalamus that should have that nickname. And this is because the hypothalamus completely controls the pituitary. So here's the hypothalamus right here. It has various nuclei in it that you are not responsible for right now. It's connected to the pituitary through this infundibulum right here. And then down here is the pituitary. And the pituitary is separated into an anterior section and a posterior section. And so these are named the anterior pituitary, or also the adenohypophysis, and the posterior pituitary, also called the neurohypophysis. The reason these have these nicknames is because the anterior pituitary is made of glandular epithelium, while the posterior pituitary is actually made of nervous tissue. And so you have nervous tracts that are directly connecting the hypothalamus to the pituitary. So the hypothalamus can control the posterior pituitary just through nerve impulses, but the hypothalamus secretes hormones to control the anterior pituitary. And then this is a chart that just shows you which hormones are released from which parts. We don't have the hypothalamus that we'll be looking at under the microscope, but we do have the pituitary gland. The pituitary gland, also called the hypophysis, when you look at it, you're going to see two main regions, and you can see separated by this border right here. The anterior pituitary will appear very dark, and this is also called the adenohypophysis. The neurohypophysis tends to stain a lot lighter because it is made of nervous tissue. Make sure that you also know which hormones are associated with each of these glands. Another slide you'll be looking at is the thyroid and parathyroid gland. Both of these are listed on the same slide. The parathyroid appears as a dark splotch on the side of the thyroid. The thyroid is indicated by many small, they look like bubblegum bubbles to me. But you see all of these circles. This is all thyroid, all of this, and even down here. And the parathyroid gland is much more homogeneous. It all kind of looks the same. The thyroid gland will have all of these circles. Later on, when you zoom in on the thyroid, you're going to see all of these circles. So this image is just showing strictly the thyroid. You can see all of the different follicles, all of the different circles. If you zoom in on one of these, you can see the differences in the cells. So the cells that make up the border of one follicle are called follicular cells. So these are the cells that directly outline the follicle right here. And you can see in the middle of all these follicular cells, you have colloid. These are the cells that are responsible for making thyroid hormone, T3 and T4. You'll also find cells that are outside of a follicle that don't make up the border of a follicle. These are called parafollicular cells. And so you find some here, all up here. Here's some more parafollicular cells down here. Any cell that does not compose the border of a follicle is a parafollicular cell. Para meaning next to. And these are the cells responsible for producing calcitonin. Now keep in mind, even though the slide has thyroid and parathyroid glands on it, this image is just showing the thyroid gland. When we look at the adrenal gland that sits on top of each kidney, there are two main parts to it. You have the adrenal cortex, which is the outside of the adrenal gland, and then you have the adrenal medulla, which is the inside. When I think medulla, I think meaty inside. Cortex always means the outer layer. When we zoom in on one of these sections, you'll notice that the cortex is made of multiple layers. You have a little bit of connective tissue, the capsule on the outside, and then you have three zonas. The outermost zona tends to stain very darkly, and it's also a relatively thin layer. And this is the zona glomerulosa. Moving deeper, you'll notice that the cell shapes change a little bit. The cells become more columnar, and sometimes you can almost see it looks kind of like stripes in there, depending on how the cells are oriented. But the cells elongate quite a bit, and that is your zona fasciculata. As you move deeper toward the middle of the adrenal gland, you have the zona reticularis. And this usually stains a little bit darker, but mainly the cell shape becomes a little bit more round. You don't have elongated cells as much as you have in the zona fasciculata. And then the innermost area is the adrenal medulla. And similar to the pituitary gland, the adrenal gland is composed of both glandular epithelium, the cortex, and nervous tissue, the adrenal medulla. So you can see that kind of duality in the adrenal gland just like you do the hypophysis. One thing that I do want to note here is to take care when you're looking at these under the microscope. In this picture, we've taken a section of adrenal gland right here. And so the order of the layers is zona glomerulosa, zona fasciculata, zona reticularis, adrenal medulla. But what if you put your slide down and you focus it over here? Now it's sideways. And you have zona glomerulosa, zona fasciculata, zona reticularis, and adrenal medulla. Or what if it's upside down? What if you take this area? It will be important to orient yourself and become comfortable with the adrenal gland. When we give you quiz and exam questions, we'll make sure you have a good frame of reference. We will not zoom in on one zone only. You'll be able to see the full cortex and at least part of the medulla. Just be careful that you are naming your zones in the correct order. Make sure at the very least you can identify the medulla versus the cortex so that you won't get confused when you put your slide down and it's not in this particular orientation. Notice the adrenal gland is kind of an oval structure. And so any part around here, you could be putting your microscope slide down and looking at any section of it. Here is one of the pictures of one of our slides in the lab. You can see the capsule on the outer layer here. The zona glomerulosa is this darkly staining layer here. And here you can see the cells kind of elongating. You can sort of see some of those stripes I talked about earlier. They're not real stripes, it's just the appearance because the cells are elongated in the zona fasciculata. And then you have another darker layer, the zona reticularis, and then you have the adrenal medulla down here. And remember, all three of these zonas are subsections of the adrenal cortex. Is an image of the pancreas. Most of the pancreas will look very similar to this. The majority of the pancreas, you can see acini, and these are the exocrine cells. So all of this dark purple stuff, these are all for making digestive juices. What we're looking for in this lab is the endocrine function. So we're going to be looking for these lighter purple areas called pancreatic islets. And these are the cells that will produce insulin and glucagon, the hormones that regulate blood sugar. If we zoom in on that pancreatic islet, you can see they look like irregular shaped lighter purple zones. Acini look very normal and regular and homogeneous. Pancreatic islets have an irregular border and tend to stain a little bit lighter than the acini. When you look at the testis, it will look like a bunch of circles. And this is because if you cut through a testis, it's full of convoluted tubing. And so basically this picture is taken, if you were to cut through that convoluted tubing and look at the inside. So you're seeing this convoluted tubing in multiple different areas. And this tubing is called seminiferous tubules. So all of these large circular areas here, here, these are all seminiferous tubules. And just like the name implies, you have sperm that are produced here. So seminiferous produces sperm. On the outsides of the tubules, you have these cells that don't compose part of the tubule. These are the interstitial cells of Leydig. And these are actually the cells responsible for producing testosterone. So cells on the outside are your interstitial cells of Leydig. When you look at the ovary, you'll see multiple circles and layers inside the circles inside the larger ovary. These large circles with the layers inside of them, and some of the smaller circles too, these are ovarian follicles. You may see some small ones, and you may see some very large ones. Those are all the follicles. The follicles house oocytes, and the oocytes are going to be very round with a very circular and even border. And the oocytes are pretty close to the same size, no matter the size of the follicle. So you can see even in this larger follicle, the oocyte is still very round. It tends to stain pretty pink versus the purple of these other cell layers over here. And it's just very round, very even. The oocyte will always be inside a follicle. And in this follicle, for example, it extends from here all the way out to here. All of this region is considered part of the follicle. All of this. And this is a large follicle. In this follicle over here, it's just this area. So this is a smaller follicle. In some of the large follicles, you'll see these large spaces. This is an antrum. And you don't need to know the antrum right now. We'll go over this slide again when we discuss the reproductive system. But be careful not to confuse it for the oocyte. The antrum often can be the same size as the oocyte. The difference is the oocyte will be very, very round, and the antrum will have an irregular shape. So just take care not to confuse those two.